All right, it's 2 o'clock. Thank you guys for being here today. I'm Matt Dean with A+ College Ready. We'll get started on this webinar. We are recording this. It'll be posted to the A+ College Ready Science YouTube channel here in just a few minutes. If you haven't already, please mute your microphone and turn off your camera. And that's just to make the process work easier, take less bandwidth. If you have a question, feel free to turn that mic back on, ask any question you might have. The PowerPoints, the Word documents, anything that I use today, I'll send that to teachers after we finish. So if you'd like a copy, just email your teacher. The next one of these webinars is going to be on the 7th of April. It'll be at 11 o'clock. And again, your teacher will get the join code and all that stuff here pretty soon. The first thing that I want to talk about today is how the exam is going to look and the new date for the AP exam. All that information was released last night. So I'm going to go through a quick presentation with you about that first. And then we'll get into the content for today. So the new exam date for biology is Monday, May 18th. And that is at 1 p.m. Central Time. I know we have a couple of folks in Alabama, like in Phoenix City, that are on Eastern Time. So make sure you realize this is Central Time we're talking about. Monday, May 18th, 1 p.m. Central Time. Here's the way the test is going to look. It's just going to have 2 questions on it. They'll both be FRQs. The first question is going to be a 25-minute FRQ. After that 25 minutes, you'll have a 5-minute time to upload your answer to the College Board. And then you'll go into the second question, which is going to be a 15-minute FRQ. After that 15 minutes is up, again, you'll have a 5-minute time to upload and submit your answer. So 45 minutes total time. That's our exam. That's what it's going to look like for us. So as far as the technology you can use a phone, you can use an iPad, a tablet, a laptop, or a desktop. They assured us in that call yesterday that any of those devices would work just fine with their website. If there are any of you that need technology, or even if you need an internet connection at home, the College Board is going to do everything they can to provide that to you. All you've got to do is go to this website, cb.org/tech. I know I've got a few teachers online today. Teachers, please share that with your students and make sure that they know that if they need that technology, they need to get that form filled out before April 24th. But that's cb.org/tech. And I think what they'll send you is probably a Chromebook or something like that. And they'll send probably a mobile like cellular internet box. So if you don't have a good connection at home, take them up on that. If you don't have a good device at home, take them up on that. They've worked hard to get those resources for you. So you're going to have 2 options as far as answering your questions. One of those is you'll be able just to type out your answers on their website. So if you're using a laptop or a desktop, that'll probably be your best option. The other option is to handwrite your answers, take a picture, and then upload those with your electronic device. Either one of those is fine. They'll both be graded the same. So you can type it, handwrite it, take a picture, upload the picture. Either one works. As far as the FRQs, the first FRQ is going to be in this format. It's going to be interpreting and evaluating experimental results. So they're going to give you some type of data, maybe a table, maybe a graph, maybe both. There'll be some kind of prompt that goes with it. And then they're going to ask you to do the following things with that information. Describe and explain some kind of biological concept, process, or model. Talk a little bit about how the experiment is designed. You might have to identify variables here, like independent, dependent variables. You might have to talk about controls. You might have to talk about another way to do the experiment. You might have to talk about problems with the experiment. all those things could come into play. You're going to be asked to analyze the data, probably make some kind of claim about the data as far as what the data mean. And then you're going to have to make and justify predictions. This is probably going to be a 4-part question, up to 10 points, and we'll go from there. But that's what the first question is going to look like, but 25-minute time limit on that one. The second question is a 15-minute one, and it's a conceptual analysis question, worth 4 points. They're going to give you some kind of scenario. It's going to describe some kind of biological phenomenon that has a problem, some kind of disruption. And what you're going to have to do in 4 parts is, number 1, describe that concept or the process. Probably going to have to explain it in a bit more detail. You're going to have to make some predictions about causes and effects of whatever the disruption is. And then you're going to have to justify those predictions based on some data. In the next few webinars, we're going to try to get some questions that look like these questions. And we'll try to practice from here on out. Every one of our webinars, we'll try to have at least 1 or 2 FRQs that go through this kind of thing. The first FRQ that we're going to look at today is not exactly like FRQ 1, but it's pretty close. What you've got to remember when you're answering all these FRQs, do it part by part. Answer part A, make sure it's clearly labeled. Answer part B, clearly labeled. Do part C, clearly labeled. Do part D, clearly labeled. Keep those parts separate. Make sure that you talk about these highlighted or bolded verbs. That's where your points come from. But we'll talk a lot more about those in the days to come. But I wanted you to know what your question is going to look like. Again, the date is Monday, May 18th. And that is at 1 p.m. Central. And there's a makeup date, but you're only allowed to use that makeup date if there's some really big conflict, and that has to be approved by your school. So if at all possible, you need to take the test on Monday, May 18th, and that is at 1 p.m. Central Time. So before I go any further, questions for me about exams, format, anything like that?
Hey, so are we going to take it at home?
Yes, it'll be at home on your own device, either your phone, your computer, laptop, whatever you have at home.
All right, thank you.
Yes, ma'am. Other questions?
Hey, if I can't take the test on Monday, what can I do? Do you know?
So you want to take it on the makeup date?
Yes, sir.
You probably need to contact. I would start off by contacting your teacher.
Mm-hmm.
They're probably going to have to talk to your school's AP coordinator, and they'll have to get that approved for you.
Yes, sir. Thank you.
But I would start with the teacher first, and then they'll probably push it on up the chain to the AP coordinator.
Yes, sir. Appreciate it.
Yeah, and that makeup date, let me put that back up. That is June 3rd. This time is in Eastern time, so it'll be 3 p.m. Central time.
All right, thank you.
Mm-hmm. All right, any more questions about the test?
All right, so how do we get to the test? Will there be like a link sent to us?
Yeah, they're going to send, they haven't discussed how they're going to send that link to you, but they will send something to you. I assume your teachers are going to have to verify that they have the right email address for you. That process is underway, and that really hasn't been shared with us yet. But as soon as I know more about that, I'll send that information out.
Okay, and will we be recorded while we're taking the test?
In terms of like a camera, is that what you're asking?
Yes, sir. Like,
no.
No, we'll not. Okay.
You will not. It's open book, open notes. You're going to have to sign something essentially saying nobody's in the room helping you. But as far as having a textbook or notes, that's fair game. looking up something on Google is fair game. They've written the questions so that it's more about the thinking than the facts. So I'm not sure how much a textbook or a Google can help you really, but those are allowable if you want to try to use them.
Okay, thank you.
Other questions?
I have a question. So the timing, if we have 25 minutes for the first question, and then 15 for the next, and then 5 minute, 5 minute, isn't that 50 minutes total?
Yeah, so I don't think their math. I don't think their math worked very well. So I guess they're saying their exam is over at 45 minutes, and then you have the 5 extra minutes to get everything uploaded.
Okay. Okay.
But it is. You're right. It is a total of 50 minutes, including that last upload.
Okay.
Any more questions? All right, well, let's move on. And if you were here for the last webinar, we ran out of time before we got to the last FRQ that we wanted to do last time. So that's where we're going to start today. This is the FRQ we're going to look at. So I'd like for everyone to take 2 or 3 minutes, first of all, and just read through it. It's a pretty detailed FRQ. So take just a minute and read through the whole thing for me, and then we'll get started going through it. All right, so this is the kind of question I could see them really putting on the exam this year because it covers a lot of territory. There's a lot of genetic stuff in here. There's also a chi-square, so there's some statistics. There's a sort of a claim, evidence, reasoning kind of component to this question. So I could see this kind of thing being there. I'm just guessing, but I think this would be a good example of something you might look for. I want to go through the question sort of almost word by word and decode some things. So let's start at the prompt. So in fruit flies, the phenotype for eye color is determined by a certain locus. That word, a locus, is really just means a location on a chromosome. Essentially, it's talking about a certain gene, a specific location on a chromosome. Big E indicates the dominant allele, and little e indicates the recessive allele. The cross between a male wild-type fruit fly. Wild-type's just the normal fruit fly. It turns out wild-type fruit flies have red eyes. So you're going to hear me talk about red eyes simply because I know that wild-type is red. You don't really have to know that. And a female is white-eyed. So we're crossing a male red eye, essentially, male wild-type, with a female white-eyed fruit fly. And the first set of babies look like this. So let's first of all look closely at that first set of babies. What I want you to pay attention to is that the males and the females are affected different ways. All of the females turned out to be wild type. All of the males turned out to be white-eyed. We've got this one weird one over here. I think we can ignore this one for right now. We'll come back to it in a minute. That's probably a mutant. But what we see is that the males and females are affected differently. And if you think back to what we talked about in the last webinar, if we had a trait when males and females inherited things in different patterns, those were almost always sex-linked traits. And since females can be affected with it, that's telling you it can't be Y-linked. So it's got to be something on the X chromosome. It's probably going to be an X-linked trait. And again, I got that from the fact that the mother originally had white eyes, the father had red eyes, but their babies, all the males had white eyes, all the females had red or wild type. So now let's look at the second set of babies and see what's going on there. So what happened in the second set of babies is that one of these females and one of these males were crossed, or probably several of them actually were crossed. And these are the babies that they produced. And notice we get a pretty good distribution of all the possibilities. We get wild type males, red-eyed females, white-eyed males, white-eyed females. So we've got to figure out now, question A wants us to determine the possible genotypes of the original parents, the parental generation, and explain your reasoning. Use Punnett squares to enhance your descriptions, but the results from the Punnett squares must be discussed in your answer. All right, so let me erase this and go back, and let's think for a second. So if we're talking about a sex-linked trait, when we're drawing out genotypes, we need to include X and Y chromosomes. So let's start off, first of all, with the P generation. Those are the original parents. The parents talked about up here in our prompt. Here's the male. Here's the female. Now remember, the male was wild type. The female had white eyes. All right, when they had babies in the F1 generation, all of the females turned out to be like the father, wild type. All the males turned out to be like the mother, white-eyed. So let's think about how that might happen. And let's just do a quick thought for a second. We've got to first of all figure out which one's dominant, wild or white. And I'm going to start off thinking that maybe wild is dominant. And I'm going to use the symbol R to show the wild type because W's are hard to tell apart. So on this X chromosome, I'm going to put a big R subscript. That means there's a gene for wild type on the X chromosome, a dominant gene. I'm not going to put anything on the Y because we're pretty sure this is an X-linked trait. So the Y is really not involved. All right. If that's the case, now let's go over here to the female. We're not sure if someone's got on their mic. You make sure your mic is muted. Thank you. We're not sure if this white one, if she's heterozygous or homozygous. Let's guess that she's homozygous. Well, let's guess she's heterozygous for right now. Because if wild is dominant, the only way she could be white would be homozygous recessive. So I show a little R, a recessive white allele on each X, like so. So if I did a Punnett square for those original parents, I'll draw that over here. Here's what it would look like. Let's put the male gametes here, the female gametes here. And what that's showing you, all the female offspring get red eyes, wild type eyes. All the male offspring get white eyes. And notice that matches up perfectly to what we had here, other than this one brown one, which we'll talk about in a minute. So I think this is the case. So our original parents, our father is going to be X, big R, Y. Making sure we know that that's a gene for wild type. You need to tell them that in your answer. And our female is going to be homozygous recessive for white eyes. If you include that Punnett square, you need to talk about it in your answer. But this is what they want us to give them essentially and explain why. Because remember, explain your reasoning. All right, so let's talk about how that works with the F2 generation. So let's say that we took one of these males and one of these females from the F1 generation, and we crossed them. So here's our male, and here's our female. And we crossed those together. So if we draw a Punnett square for that, I'm hoping you see that if we put the male gametes here, female ones here, I'm hoping that you see that this individual should be a wild type female. This individual should be a white-eyed female. This one should be a wild type male. And this one should be a white-eyed male. So we're saying that in our F2, we're expecting a 1-to-1 to 1-to-1 phenotypic ratio. So now we look at this data. And we want to see, does that look like it's 1-to-1 to 1-to-1? Well, that's what the chi-square test is supposed to do. Remember, a chi-square test is supposed to compare the results you get in an experiment to the results you expected to get. And you want to know if those results are close enough that the differences are probably just due to chance. So let me make myself a note here real quick. So we had 23 wild type males, 31 wild type females, 22 white-eyed males, and 24 white-eyed females. So we're going to do a chi-square, and we're going to see if the data that we got, this data, matches to this 1-to-1 to 1-to-1 ratio that our Punnett square says we should have gotten. When I'm doing a chi-square, I want to do it in a table form. So let me pull up a table here real quick. This is the kind of table I always want to use. Now, before we get started on the table, we want to think about our null hypothesis. And essentially, when you're doing this kind of test in biology, your null hypothesis is going to be that your expected data is not significantly different than your observed data, what you actually got in the experiment. That's your null hypothesis. And we want to either support that null hypothesis or reject it. So the first thing you're going to do when you're doing a chi-square is write in your phenotypes. Notice I use red eyes. Wherever I have red here, that means wild type. And the first thing you want to do is write in the observed numbers that you actually got in your experiment. So for wild type males, our problem told us we had 23 of those. It told us we had wild type females, 31 of those. White-eyed males, 22. And white-eyed females, 24. All right, before you go any further, the first thing I want you to do is to add up all the numbers in this column to get a total. So 23 and 22 is 45, and 31 and 24 is 55. So that's going to add up to be 100 total. So in our F2 generation, there were 100 total flies. Now, next thing we've got to determine is what were the expected numbers. What did our Punnett square tell us we ought to get for each different phenotype? And remember, our Punnett square told us we ought to get 1/4 of each of these. So the way we get these expected numbers is we take those ratios from the Punnett square and we multiply it by this total number of offspring. So our expected ratio for each of these was 1/4. So we're just going to multiply 1/4 times 100, and we're going to get 25 expected for each of these phenotypes. From here on out, we just fill in the table. So 23 minus 25 to get O minus E. 22 minus 25 to get O minus E. 31 minus 25. And then 24 minus 25, like so. Next column says to square the previous column, O minus E squared. So -2 squared is +4. Don't forget, a negative times a negative is a positive. -3 squared is 9. 6 squared is 36. -1 squared is 1. Now we want to go O minus E squared over E. Now you can work this in decimal. I'm going to work it in fraction because I think it makes it easier to do it in fractions. So O minus E squared was 4, and my expected for this row was 25, 4/25. And again, you could do that 4 divided by 25, and you could do it in decimal if you wanted to. Next one, O minus E squared is 9 over my expected 25. Next one, O minus E squared, 36 over expected 25. And finally, O minus E squared, 1 over expected 25. All right, so now we've got to calculate our chi-square value. This little piece up here is straight off the AP Bio formula sheet. So you want to have a printed copy of this in front of you when you take the exam. And I'm sure they'll send a link to you. And I'll encourage teachers to send it to you as well if you don't already have a printed copy. You want a printed copy of that formula sheet so you don't have to flip back and forth between screens. But here's the equation for calculating chi-square. It's the summation of O minus E squared over E. Remember, that little funny sigma symbol means summation. And what that's telling us is that we essentially need to add up this last column to get our chi-square value. So let's do that. Let's add up that last column. So that's 4 plus 9, 13, plus 36. That's 49 plus 1, 50. So we get 50 over 25, which is 2. Check my math again. Yes, 50. So our chi-square value is 2. Now, by itself, that chi-square value doesn't tell us much. We have to compare it to what's called a critical value from our chi-square distribution table here. First thing that we've got to know to use this table is how many degrees of freedom there are in our experiment. Essentially, your degrees of freedom are going to be your number of phenotypes or your number of possible outcomes minus 1. So we had 4 possible phenotypes. We're going to subtract 1 from that. We're going to work with 3 degrees of freedom. And unless you're told otherwise, you're always going to use the 0.05% probability value. So we want to go to where this 3 column and 0.05 row intersect. And they intersect right here at 7.82. 7.82 is called our critical value. And that critical value, again, 7.82. So we have to compare our calculated chi-square value to our critical value from the table. If chi-square is greater than the critical value, we're going to reject the null hypothesis. If we reject the null hypothesis, what we're saying is that the results we got in the actual experiment were pretty different than what we expected to get. So different that it's not likely that the differences are due to chance. But notice our chi-square value of 2 is less than our critical value. So if that's the case, which it is in our experiment, so if chi-square is less than the critical value, which it is, then you're going to fail to reject the null hypothesis. And what that means is essentially this. Yeah, these 2 sets of numbers are a little different, but the differences are small, and more than likely they're just random chance differences. So let's go back to the prompt and let's see what it says. So it says, use a chi-square test on the F2 generation data to analyze your prediction of the parental genotypes. So we did the calculations, we showed our work, and then explain the importance of your final answer. It essentially would be, because our chi-square value was less than our critical value, we're going to fail to reject the null hypothesis, which means you need to have stated the null hypothesis in your answer. The null hypothesis is essentially that the differences between your observed and expected data are not significant. So essentially, we're saying that the differences between our observed and our expected data are small, not significant, probably due to chance. We got what we expected to get. That's what we're essentially saying in part B. Questions on part B, or part A for that matter, before I move on to part C? All right, so part C, the brown-eyed female in the F1 generation resulted from a mutational change. Explain what a mutation is and discuss two types of mutations that might have produced the brown-eyed female in the F1 generation. All right, there's lots of directions you could go in this question, but a mutation is essentially any change in the nucleotide sequence of the DNA. Remember, the nucleotides are the ATCG sequences, or you might have said the nitrogenous base sequences in the DNA. Any of those would have worked. Any change in the nucleotide or the nitrogenous base sequences in the DNA. So that gave us a point for explaining what a mutation is. Then we need to discuss two types of mutations that might have produced the brown-eyed female. The two main types of mutations you probably have talked about in your class, number one, are point mutations. Sometimes you might hear these called substitution mutations. Maybe you had a stretch of DNA that was A, T, C, G, T. And then something happened, and this C got, during replication probably is when the mistake happened. Instead of a C, an A gets stuck there. That is a point mutation. It's where a single nucleotide is changed. The wrong nucleotide is substituted for the right one. In some cases, that won't be a big deal. In other cases, it can cause major, major issues. So you first should have talked about point mutations or substitution mutations. And notice it does say discuss. So you have to talk about what they are. It's when one nucleotide, the wrong nucleotide, is substituted for the right one in one place, in one location. That's why it's called a point mutation. Now, the other kind of mutation that I would have talked about here would have been a frame shift mutation. Sometimes you'll hear these called insertions or deletions. I don't want to run out of room there. So let's again go back to this sequence. So remember that when DNA is being read, it's essentially being read 3 nucleotides at a time. It's the RNA that we're reading 3 nucleotides at a time. And each set of 3 is used to code for an amino acid. What happens in a frame shift mutation is that one or more than one of these nucleotides is either deleted. So let's say that this C right here got deleted. So instead of reading ATG, CCT, CCA, what's going to end up being read is ATG, CTC. All of the codons past this mutation are going to get shifted over. And they're all going to not make any sense. They're all going to code for the wrong amino acid. The same thing could happen if instead of deleting that C, Maybe an extra A got stuck right here. And again, the reading order, ATG, that one's still okay, but now ACC gets read, and then TCC. And each set of codons is changed, and that changes essentially all the amino acids past where that mutation happened. These kinds of mutations, frame shifts, which are, again, insertions or deletions, tend to be a lot more serious in most cases than the point mutations. All right, that's it for this FRQ. Do we have questions there? All right, we're going to switch topics then. And now we're going to go a little bit into DNA structure, just a brief intro to DNA structure. And then next time we'll get into transcription, translation, and maybe even a little bit more on mutations. So just a little bit about the history first. Back in the early 1900s, scientists didn't really know what the genetic molecule was. Turns out that most scientists believed that it was probably proteins that were actually the genetic molecules. They didn't really think very highly of DNA. They didn't think it was all that important. The idea that proteins were the genetic materials made sense because scientists already knew about proteins. They knew they were built out of 20 different kinds of amino acids, and that because there were 20 different building blocks, that could explain a whole bunch of the diversity that they saw in organisms. DNA, on the other hand, they knew by this point in time also that it was only built of 4 types of nitrogenous bases. And they didn't think that those 4 building blocks could account for all of the genetic diversity that they found. But in the early 1900s, experiments started happening that proved that it was DNA and not proteins that ended up being the genetic molecule. I'm going to skip through some of this stuff. And the folks we're going to talk about really first here are Watson and Crick in 1953. Through some maybe not completely fair methods, they figured out the structure of DNA. Turns out Watson and Crick didn't really do any experiments of their own. They used experimental results from other people, a lot of which was gotten through good channels, some of which was gotten sort of through backroom channels. But they used that material and the process of just building models. They built models all day to make a model that would fit all the experiments that they knew about. And they figured it out. And what we know now about DNA, first of all, DNA is built out of nucleotides, 2 chains of nucleotides. Each nucleotide looks something like this. Each nucleotide is going to have sugar in the middle, 5 carbon sugar, and each one of these numbers is the location of a carbon in that sugar. So there's a carbon here, carbon here, carbon here, carbon here. There's a carbon there at number 5. In DNA, that sugar is deoxyribose. The 5th carbon in that sugar is always attached to a phosphate group. That right there. And then carbon 1 is always attached up to a nitrogenous base. And these nitrogenous bases are going to be either adenine, thymine, guanine, or cytosine if we're talking about DNA. But there's only one of them attached to each nucleotide. This is just one of those nucleotides. So eventually, Watson and Crick were able to come up with this model. Again, DNA is made of 2 chains of nucleotides. So here's a nucleotide, here's a nucleotide, here's a nucleotide, here's one, and here's one. What they called these pieces, they called those the backbone of DNA. And notice that those backbones are phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. And they're exactly the same. It's also the same in this side, except it's reversed. It's turned upside down. It's what we call anti-parallel. But because these backbones are the same in the DNA of every known organism, that's probably not the important part of the DNA. It's probably not the part that's the code. So eventually we figured out that the part that actually makes up the code of DNA, the important part of the DNA, are these nitrogenous bases and the orders in which they're located. Now, what holds the two strands together are hydrogen bonds between those nitrogenous bases. So these little dots represent hydrogen bonds. All of this other stuff, all of these other bonds are covalent bonds. They're strong. They're made when atoms share valence electrons. Very hard to break. These hydrogen bonds really aren't bonds. They're intermolecular interactions between polar molecules. So A and T are polar molecules. They're attracted to each other through this hydrogen bond interaction. Compared to a covalent bond, a hydrogen bond is very, very weak, very, very easy to break. Notice that cytosine and guanine are also held together by hydrogen bonds, but they're held together by 3 of them instead of 2. It's a little bit harder to break these 2 guys apart than it is these 2 guys. Now, you might think, well, why are 2 strands of DNA held together by something that's pretty easy to break? Well, it turns out that's important because we need to break those 2 apart. The 2 strands have to be separated during both replication, the copying of DNA, and also transcription when the DNA is used to make messenger RNA. So it's very, very important that the hydrogen bonds are pretty easy to break. Notice that after the DNA is put together, it's twisted into the double helix that we've all know and loved. And the inner part of that double helix are our chain nucleic, or our change of nitrogen bases, rather. So we also want to talk about the fact that the 2 strands are anti-parallel. We mentioned it a minute ago. Pay attention to our numbers here. So this would be carbon 1 here in this particular nucleotide, carbon 2, carbon 3, carbon 4, here's carbon 5. Carbon 3 is down here on the bottom. We might say, if we started at the top, that this strand has a 5 carbon sticking out the top over here. It has a 3 carbon on the bottom down here. So we might say that this strand runs from 5' carbon toward 3' carbon. Whereas this other strand, notice the bottom carbon on this sugar is the 3 carbon. And the top sugar over here, the top carbon on this sugar is 5. So we might say that this strand runs from 5 to 3. Because of this different orientation of the 2 strands, we say that the strands are anti-parallel. And that anti-parallel nature plays a big part in how replication works. And we're going to talk about replication here in a second. So again, replication is the copying of DNA. That's really, really important because it ensures the continuity of hereditary information. The entire length of each chromosome is replicated during replication. That's different in transcription. During transcription, a gene at a time is transcribed. During replication, the whole chromosome and every chromosome are copied. Replication happens during the S phase of interphase. So it happens before mitosis or meiosis gets started. It happens in 3 main steps: initiation, elongation, and termination. Now, those are sort of man-made divisions between the process, but it's a good way to think about it. And we'll talk about those 3 steps as we go. Replication starts at locations on the chromosomes called origins of replication. And you're going to see on each eukaryotic chromosome, there are multiple origins of replication, so that replication on a chromosome can happen at lots of different places at the same time. And this makes the process much faster because a eukaryotic chromosome is long. There's lots of genetic material there to copy. Whereas in prokaryotic chromosomes, bacterial chromosomes, there's only one origin of replication. And that's okay in bacteria because, number one, they only have one chromosome. And number two, that chromosome is really small and short. So it doesn't take long to copy it anyway. So here's our cell cycle. So just a reminder, we have interphase, which is made up of G1, where the cell grows and does its normal metabolic jobs, its roles. The S phase, where the cell continues to do normal metabolic roles, but also it copies its DNA. Then we get into G2, where the cell grows, some more maybe. It starts making copies of organelles. It starts preparing for mitosis. And then the cell enters into mitosis, or meiosis if we're talking about germ cells. And we go through the major stages of mitosis. Right in here would be cytokinesis, where the cytoplasm is divided. Remember, mitosis and meiosis are types of nuclear division. They divide up the genetic material, and then cytokinesis actually divides the cell in half. Here we're just seeing that bacterial chromosomes, there's only one, it's a circle. Replication happens, it starts at one location, one origin of replication. It goes in both directions around that circle until the whole chromosome is copied. Here we're seeing eukaryotic chromosomes. Notice they're more linear in shape, and that each one of these little bubbles is an origin of replication. So replication starts at all of those different locations on the same chromosome, and it goes in both directions. What we end up with are two pieces of DNA. Two pieces of DNA that are exactly alike. Another thing that I want to mention while we're here, replication is said to be semi-conservative. Semi means half. Conservative, we think of conservative as trying to keep things the way they are. When these two strands of DNA separate, each of the original strands, which are in dark blue, act as a template for replication. So each of those dark blue strands are acting as a template. And a new strand is being attached to each of those originals, so that each new piece of DNA consists of one old strand, one original strand, and one newly copied, one newly made strand. That's why this process is called semi-conservative. The new pieces of DNA consist of one original template strand and then a new strand that was built onto it. That's important because that template strand helps to ensure that you don't get a lot of mutations. DNA doesn't mutate very often. This is one of the reasons why you have a template and the process is semi-conservative. Helps to prevent mutations from happening. So let's talk a little bit about the initiation of replication. So replication starts when an enzyme called helicase attaches to that origin of replication, and it begins to unzip or unwind the two strands of the DNA at that origin. The whole chromosome's not unwound at the same time, just at that place. This creates these little replication bubbles, or sometimes they're called replication forks. And both the strands of DNA start acting as templates for the building of new strands. There are these proteins that come into play called single-stranded binding proteins. They attach to the unzipped DNA, and they help to keep it separated and also to stabilize it. There's also another group of enzymes called topoisomerases. These help to untangle the DNA because it's really coiled. It's really twisted and turned. Think of it like a knotty piece of yarn or something. The topoisomerases are essentially coming to untie it, to untangle it, to relax the supercoiling so that all the enzymes involved in replication can get to it and actually copy it. Once the DNA is all unwound and unzipped, an enzyme called primase comes in and makes these short RNA primers. These serve as the starting points for DNA synthesis. And we'll talk more about them in a minute, but they're pieces of RNA. These primers are critical because the main enzyme involved in replication, DNA polymerase, can't start a new strand, a new piece. It has to build on to an already existing piece. So that primer is the beginning of the new strand, and DNA polymerase can then build on to that. So this is a good picture for you to understand replication. So we see helicase here, unzipping the DNA. Primase we don't see here, but the primer is built. And then DNA polymerase will come in and build the DNA. It has to build the DNA in the 5' to 3' direction. Can only build it that way. So on this strand, since the original template strand is running from 3 to 5 prime, the new strand that has to be built is built from 5 to 3. Because of that, DNA polymerase can build this strand in one continuous piece. It can build it quickly. This strand is called the leading strand because it can be put together in one continuous piece using one primer at each replication fork. Whereas on the other strand, the original strand runs from 5 prime to 3 prime, which means the new strand has to run from 3 to 5. DNA polymerase can't build it that way. So as the DNA unwinds, DNA polymerase has to, or the primase has to come down and put a primer back here. And then polymerase will attach to that primer and build this way, 5 back toward 3. Well, by that point in time, some more of the DNA is unwound. So primase will put another primer here. Polymerase will come in, DNA polymerase will come in and build back from 5 back toward 3. By now, more of the DNA is unwound. So another primer will be put down, and DNA polymerase will build 5 back toward 3. So what you see is that this strand is being built discontinuously. It's being built in pieces. These little pieces are called Okazaki fragments. And eventually they have to be put together. There's a enzyme called ligase, which will actually come in and attach all these Okazaki fragments together, eventually making one continuous piece of DNA. So the elongation process is essentially what we just looked at there. Initiation is when helicase unwinds and primase makes the primers. Elongation happens when DNA polymerase comes in and attaches to those primers and brings in new nucleotides, putting them together in the 5 prime to 3 prime direction. Also, ligase comes in and connects all the Okazaki fragments together on that lagging strand, that discontinuous strand. Eventually, we get to termination. Termination happens when 2 replication forks meet on the same stretch of DNA. Not necessarily in this order, but the forks converge. All the intervening DNA is unwound, and all the remaining gaps between those forks are filled in, ligated, meaning the Okazaki fragments are connected, the different pieces get connected, and all the replication proteins, the enzymes, the single-strand embryonic proteins, all those are removed from the DNA. Again, we already said that the process is semi-conservative. Each new double-stranded piece of DNA consists of 1 original template strand and 1 newly made strand. That's important for keeping too many mutations from happening. Here we can see that happening. Red strand is a new strand of DNA. The blue strands are the original strands, the template strands. During the process of replication, there is some proofreading and error correction. Mutations do happen. Lots of times those are caught before the replication is finished, so they don't end up being a problem. There's different kinds of proofreading that can happen. I think that's beyond the scope of what you need to know for this exam, but DNA polymerase itself does some proofreading, and it can fix some of its own mistakes. There's also another type of proofreading called mismatch repair. This happens right after the new DNA has been made, and in this process, the mismatched base pairs are removed and the correct pairs are put back in. And again, I wouldn't focus too much on the details of this process. For right now, I would just know that during replication, there is some proofreading and some error correction that takes place during the process to prevent mutations from being passed on. The last thing I'll mention today are telomeres. A telomere is this region of really repetitive DNA sequences at the end of a chromosome. The reason they're important is that every time replication happens, because of that need for primers, there's a piece of the DNA at the end of the chromosome that doesn't get copied, which means every time replication happens, the chromosome gets a little bit shorter. And you don't want pieces of actual genes to be missing. So essentially, the telomeres are pieces of non-coding DNA on the end of a chromosome. You could think of them also like the little tails on certain kinds of lizards. that if you grab the lizard, the tail breaks and the lizard gets away. Think of a telomere sort of like that. It's a piece of throwaway DNA. It's there to protect the genes that matter. Scientists are really starting to look at telomeres, and what they think is that they're sort of related to aging and maybe even cell death. They think that they act sort of like a clock within a cell. And as the telomeres get shorter and shorter and shorter, the cell that controls its aging. And that when they get to a certain level of shortness, that signals the cell to go through apoptosis, which is essentially programmed cell death. So telomeres are important. Just realize they're pieces of non-coding DNA at the end of a chromosome that are there to protect the actual genes from being lost during replication. I think that's a good place for us to stop today. Any questions I can answer for you guys? All right, I'm going to check the chat, see if there are any comments there I can answer.